Good afternoon, everyone. It's really a great pleasure to welcome you to the 2014 Distinguished Faculty Lecture. I'm really happy to see so many of you here, really a great draw here. Since 1999, the Distinguished Faculty Lecture has provided an opportunity for Wellesley's most accomplished and respected faculty members to deliver a public lecture. And that helps us understand their areas of expertise. And this is an event I look forward to every year. It's one of those things where all of our faculty go out and give lectures, distinguished lectures, everywhere else, and very rarely do it to us. And so it's kind of fun to be able to hear what our great scholars are doing. So today we welcome Professor Emily Buchholtz as this year's Distinguished Faculty Lecturer. Professor Buchholtz is the Gordon and Althea Lang Class of 26 Professor of Biological Sciences. Since 1988, Emily has transmitted to students her own sense of wonder for the intricacies of plants, animals, evolution, and the diversity of biosystems. She has taught courses at Wellesley in organismal biology, comparative physiology, and anatomy, evolution, and the history of life. Professor Buchholtz's work and research lie at the intersection of developmental biology, comparative anatomy, and vertebrate paleontology. Her interest is unusual, and extinct animals has taken her to a wide range of digs and museums, and she's active in the Society of Vertebrate Paleontology. She also works one-on-one with Wellesley research students every year, and she publishes with them often. In 1996, she was awarded Wellesley's Pinansky Prize for Excellence in Teaching, really recognizing her excellence and dedication to students and her skill at bringing the sciences of life to life. Today we're fortunate to have Emily bring the sciences of life to life for us through her lecture entitled A Bush, Not a Tree, Reconstructing and Naming Life's Biodiversity. So please join me in welcoming our 2014 Distinguished Faculty Lecturer.
At the State Museum of Pennsylvania. And in that job, they said that it was an honorable thing that I was being asked to do, but basically it was to reconstruct an old collection of rocks. I was in the geology department there, and the rocks were from the Second Pennsylvania State Geological Survey. And they hadn't been looked at for 100 years. And they had been wrapped up in the 1880s and put aside, and they wanted to bring them out. So you could say that my job was a glorified rock washer. And as you can imagine, just like you, after a while I started to read the newspapers instead of wash the rocks. And because I've always loved history, and one of the rocks was wrapped in this piece of newspaper. And by serendipity, I happened to read this particular piece of newspaper. And it's on a page that also has an advertisement for Leon Perrin sauce, the Worcestershire. Delicious with meat, game, and Welsh rarebit. But it also says, honors to Mr. Darwin. It says, by cable to the Herald from London, the funeral of the late Charles Darwin will be held at Westminster Abbey on Wednesday at noon. The body of Mr. Darwin will be buried in Westminster Abbey in close proximity to that of Sir Isaac Newton. Canon Liddon, preaching at St. Paul's Cathedral yesterday, referred to the theories of Mr. Darwin as not necessarily hostile to the fundamental truths of religion. So I thought I'd start with a couple of those truths of Mr. Darwin, and you will know that he made a couple of claims. And among his claims were that species change over time, and secondly, that species share common ancestry with each other. In addition to those claims, then, he made a prediction of a hypothesis, and his hypothesis was of how that came to be, a mechanism. And he said, he named that, of course, natural selection. Darwin only included a single diagram in his entire 1859 book on the origin of species, and this is that diagram, or shall we say the left-hand side of that diagram, which is troubled by a page, the opening of the page. And so you can see right there the two things that I just said. You can see that species change in time. That's where the arrow is. And you can see that species split from each other, that they have common ancestry, and that's where I've put the red dot. But there are other lessons or other messages in that diagram as well that are kind of less explicit. One might be that species change very gradually over time, because the tilt of the line, if you call this morphology and you call this time, the tilt of the line is quite gradual. You might say that Darwin thought that the splitting of species was unusually regular in occurrence, because he's got these lines across. And you can also say that maybe he thought that all living things have a common ancestor. And that's at the bottom there, where they all come from a single point. And following Darwin, lots of people tried to actually flesh out that diagram and to put actual taxa on it. And this is perhaps the most famous. And it's by Haeckel, a very famous German evolutionist. And you can see the year is 1866, so this is just a few years after Darwin wrote. And you can see that this is supposed to be a history or a pedigree of man. And you can see that much more than Darwin even, once you start putting names on a diagram, you start reflecting kind of the knowledge of your culture, the time and the culture that you live in, so that there are things here that might embarrass us today a little bit. For instance, you'll notice over here he's calling this primitive animals. Well, that's a word that we don't use anymore. We never say the word, we try very hard not to say the word primitive. You can see that although the species are splitting from each other, that the organization of this is very vertical, that there are animals down here at the bottom which are considered primitive, and they lived maybe a very long time ago, whereas those animals that are considered very specialized live today or at the top. And you'll notice that there's pride of position on this chart, and that you are there. Next to the orang and the gorilla, and then underneath them the semi-apes. If you've ever asked yourself why the order of mammals that we belong to is called the primates, don't think any further. There's a reason that we are called primates. It's because of a self-conception. Now, what might not be quite so easy for you to recognize, especially if you're not a biologist, is that this tree is divided into what we call prokaryotes and eukaryotes. And eukaryotes are those organisms who have cells that are quite large. They have an internal cytoskeleton. They have organelles. And they have a membrane-bound nucleus. So that's the picture on the top. And all of them in the red circle here are eukaryotes. Whereas the prokaryotes are much smaller, like 1/10 of the size in cell diameter, and they don't have that internal structure, and they don't have a membrane-bound nucleus, and they are restricted down here in Haeckel's view only to the bottom of the chart. If you were trying to identify who you would call a prokaryote and eukaryote among the organisms you know, only the bacteria would fill this category of Monera, or prokaryotes. Okay, so what, I think I've missed something. Well, I'm just going to say that we don't think that the history of life looks like this anymore. And the reason we don't is because of a couple of innovations or tools that we use, and the tools we're going to use are: one, the reconstruction of the branching sequence in the history of life. We think we now have a way to actually predict what that branching sequence was. And second, that we have a much better idea of what the origin of morphological variation is in the developmental process. So I'm going to go through those two big points there, and then I'm going to give you some examples of what we're calling developmental paleontology, which is the kind of work that I'm doing now. Okay, so I'm going to start here with reconstruction of the branching sequence in the history of life. And so this is going to give you a little tool that you can use. My students are laughing because they've seen this picture before. This man's name is Willy Hennig. He was a German insect taxonomist. So he spent the 1960s developing a way to try to reconstruct the branching pattern of life. And so we're going to do this with 3 animals because Hennig said that you could always tell, you could determine which 2 of any 3 species were more closely related together with his process. And so the 3 species that I have for you today to try to tell me who are most closely related, or to use a biologist's word, which 2 of the 3 have the most recent common ancestry, which is how we define most closely related, are going to be a shark, a bony fish, and a chicken. And if you think I'm setting you up, you are absolutely right. I mean, there's no way that I'm not setting you up, right? All right, so if I were setting up a naive, if you were completely naive about this, or if you were using common language of the day, you would say that well, there's fish and there's birds, right? And so the fish must be more closely related to each other than the bird is. What Hennig said is that what you're using is some kind of overview of similarity. And unfortunately, similarities come in 2 types. And only one of those types of similarities is usable. So in Hennig's view, any similarity that was extant at the time of the common ancestor of all 3 of those taxa, which would be at the red dot, is unusable. And the reason that similarity is unusable is that all 3 of them had perfect access to inherit the traits that were at the red dot, at the common ancestry. But we're actually looking for traits that evolved after the red dot that were on the lineage that led to 2 out of 3. So anything that was there at the red dot is completely unusable. So you might say, well, those 2 organisms, those 2 animals on the left share water habitat, they share gills, they share a certain number of fins or something like that. And the question you would have to ask yourself is, were those present at the red dot? How can we possibly know whether they were there at the red dot? And it turns out that Hennig proposed that we use what is called an outgroup. That we take an organism that we know is outside this group, and again, I suppose that is disprovable. It's a hypothesis that it's outside the group. But one that is far enough distantly related that you're pretty sure it's not in the in-group, as we call it. In this case, what I've used is a lamprey, which is a jawless fish. And I say to myself, well, does it live in the water? Does it have gills? And the answer to those questions is yes. And because the answer to those questions is yes, it means that I cannot use those traits to try to predict which 2 or 3 out of the 3 of these are more closely related. So it turns out that when I do this strictly, I come up then with something that looks like this, which says that chickens are more closely related to one group of fish than that group of fish is to other groups of fish. Yes? And that's because I should have put another colored dot right here, that there would have been a point there where some traits evolved that are shared by chickens and bony fish that are not shared by sharks. Well, this is a perfectly kind of counterintuitive result, right? And it turns out that when we do it over and over and over and over again, we come up with some very surprising results about who is most closely related to each other. It also has a wonderful other little advantage here, which is that it allows us to sequence the events in the history of life. Because we know which those, what we're going to call derived or non-ancestral traits are, we can actually put them in a sequence. So if you would have come in here naively today and I said, which evolved first, feathers or gills? Hey, but now you know, right? Gills evolved first and feathers evolved second. So we can actually make a sequence of events of when traits evolve. This thing works so well that other disciplines have taken it over from us. This is for Cappy.
You've sent this to me.
Yes, I have sent it to you. But it turns out that you can use this with anything that has a genealogy to it, that descended from an ancestral form and then derived from it, or maybe there are many, many descendants and you're not sure which of those are most closely related. You can do this perfectly well with in other fields. They said this talk is supposed to have a liberal arts application. Well, this is it. This is it today, all right? You can imagine that when we do this, when we build up the branching of life, and I promise you there are hundreds and hundreds and hundreds of people in different taxonomic groups doing this as their job every day, trying to figure out what the tree of life looks like in their particular discipline, you can imagine that we might get a tree that looks like this, or you can imagine any number of other possible trees. And I'm going to tell you that we don't get trees like this. We get trees that look like this. Okay, what is it about that? How would you describe that tree? And for me, the characteristic of that tree that's most telling is that it's got a very wide bottom, and that the original the diversity of the whole group is there almost from the very beginning. Yes, there are groups that are diversified within each of the branches of that wide radiation. So there is increasing diversity, but I'll say it this way, the disparity there is there right from the beginning. The ways to be an X are present right at the very beginning and then radiate. We don't really often know what it is that makes an adaptive radiation happen, but basically we think it's an interaction between an ecological opportunity and some kind of biological innovation. So that's my transition to talk to you about, well, how does that innovation come to be? If we have the ecological opportunity, which when we talk about some of the more specifics later, I'll be trying to tell you what I think some of those opportunities might have been. But here, it's going to be, we're looking at how the diversity that must lie somewhere here at the base of the adaptive radiation, how those came to be. Okay, so I'm going to take you to the process of development. In the process of development, you're probably very familiar with that top rung of what development looks like. That morphology emerges over a period of time after the fertilization of the egg and the zygote over here on the left, and that embryos at different stages of their growth look differently from each other. But maybe you've never asked yourself, what's going on at a cellular level? So I'm going to take you from this zygote here to that pancreas cell there and suggest that what is happening on a developmental layer that you can't see on the cellular level is that the DNA is actually being replicated exactly each time so that the DNA that's in my hand cell looks exactly like the DNA that's in my pancreas cell. There is exactly the same genome in every cell. But clearly, different parts of that DNA are being expressed because my hand cell doesn't look anything like my pancreas cell. So I want you to think about development on a cellular level as being a mechanism by which DNA is being restricted, by which I mean that a smaller and smaller subset of the entire genome is being expressed as development proceeds. So whereas in the beginning, it's possible for that cell, you've heard the word stem cell, certainly, it's possible for that cell to become any cell. As development proceeds, more and more parts of that DNA get shut down so that the possible futures of that cell get restricted over the course of development. So what we can actually do is we can map the developmental process. So this is, I just stole this from the literature. But I'm going to ask you to think up there at the fertilized egg at the beginning and know that the genome that's in that is still completely accessible to any cell that's descended from it. But that as soon as we have a split there between AB and P1, which was a part of the article and is not pertinent to the discussion here, that those cells are no longer omni-capable, but rather they have a subset of their genes able to be expressed. And that as development proceeds, we can imagine that happening over and over and over again. So that by the time you have a cell in your hand or a cell in your pancreas, it's only expressing a very tiny subset of the genome that is still present in the nucleus. But it has differentiated physically, and it has been determined by which DNA is still accessible. If you look at this pattern, we would call it hierarchical. And as it turns out, another word for that is modular, because then from, say, this EMS, there will be a module of everything that is descended from that, or there'll be a module that's everywhere descended from this. And because development is modular, it turns out that morphology, that anatomy, is modular as well. That anatomy is the product of some subset of each one of these modular units of development. And you can imagine that when mutations happen, that they happen to development. We like to say, well, mutations happen to the organism, but they actually happen to some part of the developmental process, because morphology is the product of development. In fact, we even say that evolution is the product of development changing over time. So the way that morphology is generated changes slightly over time, and that is we see as phenotypic or morphological evolution. So I've put different colored dots on there. The dots are mine. And you can imagine that if a mutation happened early enough in the developmental process, by which I mean perhaps the green dot up there, that would eliminate the possibility of a huge amount of that genome ever being used, and that embryo would die. And you can imagine that if the mutation happened in the developmental process at the very, very last stages of the development, that we might have a finger that's shorter than another one, or something, some minor difference, but that would not be a mutation that would allow an organism to move its adaptive zone or be the base of an adaptive radiation. But that if a mutation happened that were somewhere in the middle there, you can imagine that the organism still might live, but it might have different enough anatomy that there might be some kind of mutation that it would allow it to invade a new adaptive zone or be the base of a new radiation. So the trick is, where in the developmental process do mutations arise? This is an adaptive radiation, and I've suggested that early on in an adaptive radiation, there's a mutation that's big enough to make a difference in how the morphology of that animal exists, but small enough that the animal didn't die, right? That there's a significant change in the morphology of that animal. And that later on in an adaptive radiation, later on in this unfolding of the diversity of life, we can imagine that those mutations are smaller and smaller and smaller. And this was said best by someone other than me. This is Charles Marshall at Harvard. And so I put his quote here. He says, "As evolution proceeds, the developmental stages responsible for laying down the body plan are overlain with downstream genetic pathways, making it progressively harder to modify those earlier pathways." What this means is that evolvability changes over the time of an adaptive radiation. The opportunities are there right at the beginning, and that the rest of the adaptive radiation is playing out the themes that were already laid down at the very beginning. This also means, strange to say, that evolution is constrained. We used to think, in fact, we used to have a little computer program where we could change any structure we wanted, and we would see how we could draw the organism over this direction or this. It was great. But it turns out that it was a poor representation of evolution. It was poor because the process of adaptive radiation itself is constrained by the innovations that happen in the process. So that we like to think of evolution as being particulate. Say there was an animal, this very bad word, you never should use it when you talk about evolution, needs a longer whatever, or a bigger or whatever. We don't say that. But say a longer finger would be adaptive. It turns out that sometimes these changes cannot happen without changing other things too. And that is because of that developmental hierarchy. Because there's a whole module, if you change one piece of the module, unless it's a mutation that happens at the very, very tip of the developmental process. But if you change anything that's up in the middle of a developmental module, you're going to change a lot of other things as well. And those things might or might not be adaptive, so that there become things that cannot evolve. There are constraints that evolve. All right, so I'm going to show you an example of an adaptive radiation and of what we are now calling developmental paleontology. So I'm going to say that developmental paleontology is the study of how the developmental process constrains the unfolding of the history of life. And this is, Nigel Hughes was probably the first person to kind of put it all together, and his study was with trilobites. So the first thing I want you to notice is that his adaptive radiation of trilobites is exactly what I've just described to you. That there's this burst of evolution that happens right at the beginning of an adaptive radiation, and the distance from here to here is as big at the bottom as it is anywhere in the chart. So all the basic ways to be a trilobite were there in the Cambrian period, which is a really long time ago. And then within the groups that originated the different ways to be a trilobite, we had tweaks on the system so that they became a little fancier, a little this or whatever, but basically they were all there at the present. And we're not sure what the developmental innovation was that would have been right there at the beginning, but almost certainly it has something to do with the way that the different segments of this arthropod were fused together into the cephalon at the top, and how they were free in the posterior part of the body. But it turns out that as trilobite evolution proceeded, what we notice is that within each one of these groups, we start having specializations of particular parts of the body. And so I put one of the derived trilobites up there at the top. And unfortunately, every time you introduce an innovation, you are making a submodule of your developmental process, and you are constraining further evolution. So that means that if the environment changes radically enough, what happened to the trilobites may happen to you, right? Which is that it is no longer possible to change. The constraints limit the evolution that is possible at that time, and the group goes extinct. So in some kind of ways, evolution is its own. The process of evolution limits itself because of the specialization within the developmental program as evolution proceeds. Okay, so I'm going to give you two examples of the projects I've worked on with my students. And the first is here with the evolution of mammals. And so you will know about mammals, yes? You know mammals, and you know we're very special. We're right at the top of the tree, right? Yeah. And we're a very diverse group of animals, and so I put here some of the traits that we associate with mammals. Live birth, hair, mammary glands, 3 ear ossicles, you may not, that hammer, anvil, stirrup that you memorized in grade school, and a muscularized diaphragm. Lots of animal groups have diaphragms, lots of vertebrate groups, but not a muscularized diaphragm that is an aid in ventilation of the lungs. So we'll say that there's lots of specializations that mammals show relative to their outgroup, relative to reptiles. But what you may not know is there are also constraints. So what I've put up here is a picture of a Mesozoic reptile, which is a plesiosaur, which, as you can see, has an impressive neck. Okay? So in all other groups of vertebrates, if you need a long neck, the way you do that is you add vertebrae to it. You become long neck by adding vertebrae. And it turns out that mammals can't do that. So the famous constraint that was recognized at the time of the French Revolution, and people have been wondering about ever since, is the fact that mammals have set numbers of vertebrae in the different parts of their body. So if they want an elongate body, what happens is this. I've charted here the length of the vertebra of many different carnivores, which is one order of mammals. You can see that basically what they can do is they can elongate those vertebrae, but they can't add vertebrae. And most particular of these numbers, this is the one here that is most tightly constrained. 7 cervical vertebrae in all mammals. So we wanted to explore that and find out what had initiated, what had made that constraint in the evolutionary history of mammals. And so the first thing we do is we build a phylogeny. We use Mr. Hennig's tool, which is try to reconstruct and use the work of many other workers, not just ours, but put the traits that we're most interested in on that phylogeny to see if our constraint evolved at the same time of any of these particular innovations that make mammals who they are. Because we're convinced that our innovation must have happened very early, because all mammals share it, and we're convinced that it must be tied to another innovation that's very important to mammals, because if it weren't very important to mammals, you would think that they would have let their neck vertebrae change count at will. But something is holding it back. Which of these innovations is holding it back? And so it turns out that our innovation coincided in time with only one of these, and that is the muscularized diaphragm. So then what we do next is we go to the developmental process in living mammals to try to see if we can make a connection between the development of the muscularized diaphragm and the development of the muscularized the muscularized diaphragm with a stability of count. And it turns out that the somites here, I'm looking at the back of an embryo right here, and you can see that it's got these little blocks of mesodermal cells. These are going to be the source of the muscle cells that are going to travel to your forelimb, to your hind limb, actually to the base of your tongue. And if you look at the embryo on the side, this is a mouse, you can actually see the streams of cells coming out to the tongue, to the forelimb, and to the hind limb. It would seem like those cells should already be out there somehow in the limbs, but it turns out they migrate during embryology out into the forelimbs. And there's a cartoon of them coming out of the somite to migrate into the forelimb. And it turns out that the muscle cells that migrate into your diaphragm come out from the somites that are immediately adjacent to the forelimb. And some of you may know that the diaphragm is innervated. The nerves as well as the muscle cells come from the neck region, because you know if people have an accident that actually truncates their spinal cord somewhere above the diaphragm, they can still breathe. And the reason they can still breathe is that it hasn't, that these nerves are still intact up here from the neck, and the phrenic nerve comes down to innervate the diaphragm. So we made a hypothesis, and our hypothesis was that in non-mammals, here are the cells coming out to enter the limb. The forelimb could move at will up and down the column because it wasn't impeding any other module of development or morphology. But here in a mammal, because those cells up here had already been committed down to a diaphragm, free movement of the forelimb was now constrained. And the rest is more details than you probably want to know about that. But basically, because the source of cells is the same, we can no longer use those cells for two different functions. We can no longer move the forelimb relative to the rest of the body. Well, that's a very nice hypothesis, but you remember that we try to test our hypotheses too, right? Okay, so it turns out we can't go back in the past and find out whether this originated at the same time, but it turns out that I lied to you, that there are 3 mammals that break the rule, okay? So we can look at those 3 mammals that break the rule and see if our predictions hold, and those predictions happen to be from details of the developmental process that I don't want to get into here. But basically, we would predict that if you did change your vertebral count in the neck, that it would lead to a poorly muscularized diaphragm. And because the forelimb and the hind limb are part of the same developmental module, if one changed, the other should change as well. So this is a two-toed sloth, Calepus. Very famous animals, famously said, if one more thing were wrong with this animal, it would cease to live. And what we did is my students and I traveled around the country to museums. Michelle, raise your hand. Here she is. My previous student, who now works for the college, measuring and dissecting. And it turns out that we were right. Okay, so hey, it turns out what I've got here is the pelvis of two-toed sloths, and these are lumbar vertebrae. And what I want you to see is that the pelvis has been shoved up onto the lumbar vertebrae. And the lumbar vertebrae are in various states of distress. Here they're just separated from the pelvis, which is unusual. Here they're saying, no, I will not fuse with you. There's a gap there. And in this guy over here that we found at the Field Museum, they're actually trying to escape from the pelvis. All right, so it's a very nice confirmation that we were onto something, that this was a modularly organized thing and that the two traits were tied together. A project that we're just in the current state of developing has to do with whale evolution. And you will recognize those as whales because they don't have a pelvis and they look elongate. But what I want you to look at is the fact that one of them has double the number of vertebrae as the other one. That basically they're both the same length, but one has double the number of vertebrae. So it turns out that there is a dolphin pattern, and that pattern basically probably for fluid dynamic reasons, a dolphin can't really change shape. They all look about the same, even if of slightly different size, but they all have the same shape. There's not elongate and short dolphins. And we want to know how it is that this innovation could occur and allow dolphins to invade a new environment, an adaptive radiation of dolphins that happened in the Miocene and allowed them to go into the kind of cruising predators that they have been in the open ocean. And so the first thing, again, is we look at our phylogeny and we identify where on the phylogeny these innovations happen. It's way down here. You can see all the dolphins up here. And then over here we have things like sperm whales, and then over there still more fossil whales that have deeper diving, but not that cruising predation that happens in the open ocean. And then we look to the developmental process to try to see if we can find something that tells us why whales have been able to do something that nobody else has done, which is to double the number of vertebrae, and because of that, probably build a vertebral column that's half vertebral disc and half bone, and allow a great flexibility for cruising so that they have a lot of elasticity in their vertebral column instead of just bone. And our hypothesis, not proven, not tested yet, is that they have been able to dissociate two important processes of developmental biology. One of those is how fast you make a somite. And so again, somites are the muscle blocks that exist up and down the back of an animal. And we know from the molecular biologists that the speed of that process of making somites, as somites are laid down, determines how many vertebrae you will get. But at the same time, usually what happens is that the axis elongates too, so that large numbers of vertebrae are associated with long animals, like a snake. But here we've got something else happening. We've got a large number of vertebrae in a relatively fixed length animal. And so we think that those two processes must have been dissociated. How can we test this theory? Well, it turns out that even though whales lack a pelvis when they're adults, that as embryos they have hind legs. And if you look at this embryo over here, that H is the hind limb of a very early embryo, and you can see that it's basically disappeared by the time the embryo is just a little older, whereas the forelimb has gotten bigger. And so the way we're doing this is that we are finding embryos of whales at different museums around the country, and we CAT scan those embryos in the hope that we can identify where the sacrum is. Because it turns out, very strangely, that again, details of our hypothesis, which are too detailed to go into here, predict that the sacrum of whales, I know this will sound completely counterintuitive, actually exists in the middle of the lumbar region. I didn't think it up myself. It turned out that in the 1930s, a very famous man, E.J. Sliper, on the basis of morphology alone, suggested that the sacral vertebrae lay in the middle of the sacrum. And people wrote it off, and I bet he's right. But in any event, what we're doing is we are CAT scanning whale fetuses. That's one up there. And then what we're doing is we're using a very sophisticated computer program that allows us to take each subunit of that skeleton and figure out how ossified, how dense it is. And so this is the computer program saying this is this density, and separates and gives us a nice chart about it. Because we know that in other mammals, density changes by series, by thoracic, lumbar, pelvic. They all have different densities early in development. So we're hopeful that we can reconstruct where that sacrum used to be and see if Mr. Sliber and we are right in our hypothesis. All right, so you could say that I'm dealing with a little tiny piece of the adaptive radiation. I'm dealing with the adaptive radiation of whales in one place, or of the base of the mammal tree in the other. And with other people, we hope that we can build a developmental paleontology that asks us to ask kind of meta questions about paleontology and the history of life. Questions like, are there certain kinds of developmental innovations that always happen at the base of adaptive radiations? Does adaptive radiation always come with constraints? Later in development of a phylogeny, do we have different kinds of mutations that occur at different times? These are all questions that excite me a great deal and that help us reconstruct what the tree of life looks like. But we do know already that the tree of life doesn't look like Mr. Haeckel's tree of life, and in particular, that it is horizontal in organization. And in particular, I want to say that complexity evolves in each area of the tree of life. There's no such thing as complexity at the top of the tree of life and primitive at the bottom of the tree of life. Rather, complexity, we have huge branching, and complexity evolves in this group, and this group, and this group, and this group. Also, other animals are not, like all these animals on Mr. Haeckel's chart, these animals are all alive today. And here on his chart, it looks like they came previous, and then that man was the crowning evolutionary event at the end. But rather, we are embedded in other complexly evolved animals. We are not separate in kind. We are not more complex. We are differently complex. So if I take Haeckel's colors again and try to put them on a phylogeny that was generated by someone who works on the molecular level, this is the phylogeny here, and I put the same colors on it that are on the chart over here. So all of these eukaryotes are in this little part right here, and all of these prokaryotes here, we can open up the circle and have it lay out flat like this. You could see that almost all the diversity of the world is prokaryotic. That's where the metabolic innovations came. That's where the early plans of how to live on planet Earth with its constraints and its ecology were developed. And we are Johnny-come-latelys and a very small part of that radiation. And then if you just want kind of the take-home message about what does this mean that we're embedded in the tree of life, what kind of biological humility do we need to walk out of here with? I want you to look here and see that at this junction right there, the eukaryotes are most close relatives of one kind of prokaryote, and that they are then more distantly, both this kind of prokaryote and the eukaryotes are more distantly related to this other great group of prokaryotes. So this is what it looks like. Thank you very much. Questions? Kim.
So if you back up a
Oh.
slide or so,
Okay.
does this mean that 5 billion years from now, right before the sun dies and really toasts everybody, that the dominant form of life on Earth you think will have come from some other distant part of the blue circle?
I think that all 99.9% of all species have gone extinct, and we will go extinct. I do think that life will continue on the planet, so I guess the answer is yes.
But I guess, given what where the prokaryotes and eukaryotes sit in your circle there, do you think that the dominant form of life in the future will have come from one of those other blue branches?
Yeah, your question is very interesting because it asks basically, where do new innovations come? If everybody is complex already, where does new complexity come from? And it typically comes from the base of a not too specific part of a previous adaptive radiation. So it won't come from the most complex or the most integrated genomes or the most complex development at the end of a radiation, but somewhere near the bottom. And so it would come from a very early rodent, or it might come from a very, something like that, something that had wide ecological tolerance, because we're changing the ecology of the planet so much that I can only imagine that it would come from an organism that had really broad tolerance. Cockroaches, another good example, right? Anybody else? Yeah, Dan?
From this perspective, it seems like mutations are driving much of that process, and that I'm fascinated by this notion that you get this sort of loss of degrees of freedom as you go further up.
That's a good way, yeah.
But another driver, which is recently getting a lot of attention from the human public health perspective, is epigenetics. And so the plasticity of the DNA sequence post-embryo and in early life. And what do you think the interplay between that driver is and the more embryonic mutation driving evolutionary pathways?
I'll have to dance places I don't usually dance, but my guess would be that it would be a much smaller magnitude. That epigenetics would only allow us to express a little more, a little less, greater tolerance, less, but it wouldn't be able to change our morphology in the profound ways, probably, that a mid-level developmental sequence mutation would. It would, yeah. George? Well, epigenetics is basically what happens that is not controlled just by the gene, but in controlling how it's expressed after it's in the organism. Okay, so it's kind of the post-inherited expression of that gene. Marianne?
I think I know the answer to this question, but as you move up the tree or the bush, and you're losing those degrees of freedom, what's triggering those adaptive radiations that occur near the tips? I'm thinking of Darwin's finches, which is the classic example of adaptive radiation that we teach in Bio 111, and so it's not a change in development; it's probably a change in behavior.
Allele frequency, certainly. Certainly a change in allele frequency. I still think you could call it, it would certainly be on the scale that I'm representing here. It would be a very small twitch toward the top of a very specialized kind of bird that lives a particular kind of way. It would be a minor adaptive radiation that's within a much bigger radiation that is birds. Would you have called it another way?
No, I guess I was just suddenly thinking, wow, were all of these adaptive radiations triggered by changes in development? And of course they weren't. I don't think.
Well, it depends on how broad your definition of development is. But basically, if we see development, morphology as the product of development changing over time, then they do have a developmental origin. But I think like the Darwin's finches would be something that's in the very late stages of the development. It would be how much kind of growth enhancer you have for the size of the beak. Here, it would not be beaks versus jaws or something like that. It's a more terminal tweak on the system.
Right. So you would argue there is a developmental component there.
Definitely. Who's it? Go ahead. Mala.
my best guess is that would have only happened in the very early history of life when these systems were evolving. The gene checking systems have been in place for, I mean, they're so broadly distributed that we must assume that that's how we define things that came, when we need a quick way to say when did something evolve. The best way is to, if it's broadly distributed, we think early, that it was early. And because those mechanisms are so broadly distributed, I think they must have been in place early. And I don't think there's changes in that within recent, more minor radiation, because we can look back at the very beginning of life and say, well, that was the greatest adaptive radiation at all, right all the way at the bottom. Yeah, Roxanne.
Thank you so much for this. I've learned a great deal. And so my apologies for asking a question out of left field, literally and figuratively.
I was going to say you are a political scientist. Yeah.
I ask it with great humility. You know, given that in the 19th century there were a lot of philosophers that tied together a certain kind of species arrogance with a certain kind of civilizational arrogance and tied it to a moral arrogance that justified all kinds of political asymmetries and grabs for power and land, I'm wondering if you might think with me if there are any ethical implications to your argument essentially for a biological humility. In other words, are there any ethical imperatives that flow from that kind of paradigm shift that you've outlined for us?
Absolutely. Thank you so much. I think it has something to do with how we treat the rest of the creation, that they are as entitled and we are embedded in them, that there is no biological hierarchy in the vertical sense. We are only co-inhabitors. Absolutely. Thank you. That's, yeah. I don't know if I would be willing not to take antibiotics when I'm sick, but yeah.
Talk about humility. I hope this isn't too much off topic, but microbiome. I'm here to make my microbiome happy. Otherwise, I'm not here. And I'm just curious, how much of the collective me is eukaryote as opposed to prokaryote?
Well, so I had to actually chop 2/3 of, you can imagine how much I had to chop out of the talk. But it turns out that a very, very large percentage of you is prokaryotic, because the metabolic diversity that existed at the very beginning of life are split into 3 kingdoms. Those are actually 3 domains. These are eubacteria, archaea, and eukaryota is how we name those domains today. But it turns out that the diversity that's within the 2 prokaryotic domains there, the eubacteria and the archaea, are what keep us running. That those cells that know how to do things like use oxygen to make ATP, for those of you who know those words, we have taken them inside our own cells and let them do their work there. So we are colonies in 2 ways. We're colonies within each of our cells has been colonized by mitochondria, who are prokaryotes. And then, of course, the microbiome in your gut you live maybe for and with, embedded with that, and we would not live without either. So there's many more cells of prokaryotes in this, what you see in front of you, than there are cells of eukaryotes. Yeah. Mm-hmm. They are. Well, I'll give you kind of, I mean, it's a profound question, and I'll give you the part of the answer that I give in Intro Bio, which is because I don't know as much as you know about plants, which would be that plants have a different program of differentiation and shutting down the genome than do animals. They tend to keep much more of the genome plastic much later in life. So there's very few cells that are terminally differentiated. A flower is terminally differentiated. It cannot give rise to a root or a stem or whatever. But most of the other cells of a plant, you know yourself from taking cuttings, right? That you could take a stem and stick it into potting soil and you get roots, right? You can't do that with an animal. You can do that with a plant. So so I do think that there's a shift in how that develop, how much constraint and how much not in plants. Yeah. But I think the mutations could come at different places to give bigger jumps than with animals. I think animals shut down earlier. They are differentiated earlier, determined earlier. Yeah, it's a great question. Carol. Say it again? Yes. Mm-hmm. Well, certainly most species have become extinct. But I think I'm missing one connecting logical piece. I don't predict that all species will become extinct at any one time, but rather that different species will come forward. And that it isn't because different adaptive radiations are at different points in their adaptive radiation. It's quite possible that some humble organism that we don't look at right now will be the source of a new one and start kind of at the base of an adaptive radiation again, and those would live on for many millions of years. But I don't predict that all taxa are now approaching terminal complexity or anything like that. Is that kind of what you meant? Yeah. Oh. I've never thought about that. You mean by man's change of the plant? Yeah. Yeah, I don't often think of that little tiny time slice that's at the top. I tend to think, yeah, tend to think more deep. But certainly we are changing the environment rapidly. And if, for those adaptive radiations that are close to their complexity, adaptability limits, those will be the ones that are most vulnerable. And those without such fine-tuned adaptations will have a better time of it. I guess that's all I can guess about that. Yeah. Yeah, David.
I've been impressed over my career how much our knowledge of how some aspects of the world work changes. And our understanding, the progress of science. If you were to give a similar talk 20 years ago and have that beginning part be a summary of this is our understanding of evolution, how different would that talk be?
It would be very different, actually. The two revolutions I talked about, the developmental biology and the phylogenetic systematics, are absolutely integral to this. And when I was in grad school, in fact, I brought it. And when I was in grad school, the first war was going on. And this is my old copy of phylogenetic systematics, because the young bucks, of which I was at that time, we thought that the old guys just didn't get it, and that we had to have phylogenetic systematics. The developmental biology has been much more recent. And so trying to integrate those two together, Nigel Hughes, his first Trilobite papers came out like in the year 2000, 1999, 2000 or something. So putting these pieces together across disciplines is what's so exciting about it for me. And I think I think it wouldn't have existed. It did not exist at the beginning of my research career. Yeah. Hello, Jocelyn.
So I have a non-sciencey question for you, mostly based on your experience about this. But I've been recently reading about the Dover education case of teaching evolution in schools, and I'm not going to go too political.
Thank you very much for not going. Remember, not necessarily in conflict, right?
Right.
Back in 1882. Yeah, right. Yeah.
So one of the conversations that you and I had was that it's tricky to make this concept accessible for those of us, not even maybe all of us in our department of biology, but even to go as an evolutionary biologist to make it more accessible in a liberal arts setting. And I think, we all made statistics about how many people don't believe that this is actually going on, and that maybe scientists are not communicating this
Mm-hmm.
effectively. And my question for you is, has this talk and maybe developing it in a way that you can communicate it more accessibly given you any ideas about how we can communicate our ideas about evolution and how it occurs? That might be too political for you, but
It is a little political, but Roxanne, you can help me with it.
it's really just more how do we, have you thought about as you're doing this?
I don't. Okay, it's not going to be a very nice thing I say. Okay, but I don't think that you can convince people about evolution by rationally having a discussion with them. I think in many cases the concept of evolution that people hold is a part of a worldview that's integrated with other things that are held so close and so dear that when you use rational, what you are actually saying to this person is, you don't count. You look at the world wrong, or something like that. And I think that's an unacceptable message. I'm reminded of Ernst Mayr, who those of you who don't know is the great evolutionist of the 20th century, lived to be 104, he was at Harvard. And toward the end of his life, he used to hold these, I don't know what they were, awe sessions where he wouldn't speak, but he would sit in a chair on the stage and people would sit in front and ask him questions and he would respond to a particular question. And I was at one of these and someone said to him, well, Herr Dr. Professor Meyer, if someone said to you, you were on an airplane and you were next to them and they said to you, I don't believe in evolution, what would you say to him? And he said, and he brought the house down, and he said, I would say, have a very good day. Yeah, Vanya. Mm-hmm. Yes, and I suspect that they will not be very different from each other. Identical, yeah. Huh. Another vote for great diversity within prokaryotes or basal eukaryotes, yes? Yeah. Mm-hmm. Okay, so this is our Vanya's our new microbiologist. Thanks. Thank you, everybody.